Hello, everyone. My name is Zhang. I'm a PhD candidate in the Faculty of Engineering at Macquarie University and the Respiratory Technology Group of Woolcock Institute to Medical Research. Thanks for DDL give me this opportunity to present one part of my PhD project, an adaptable dual-chamber microfluidic platform to investigate the Pseudomonas aeruginosa biofilm growth at air-liquid interface on the controlled hydro and aerodynamic flows. It has been estimated that 65 to 80% of human infections are associated with biofilm, with Pseudomonas aeruginosa biofilms being the major cause of hospital and ventilator-associated pneumonia and responsible for many chronic lung infections. The reduced efficacy of inhaled antibiotics is due to the presence of bacterial biofilms. The biofilm formation starts with planktonic cells, also known as active cells, finding attachment sites, such as the lung epithelium or mucus layer, to form irreversible attachment. And at this stage, they are still susceptible to antimicrobial agents. Then the bacteria rapidly proliferate, forming microcolonies by producing extracellular polymeric substance as a protective layer. Subsequently, the biofilm matures and becomes super resistant to antimicrobials. And the cycle continues with biofilm detached, whereby planktonic cells colonize other areas in the lungs. Once the biofilm is established, these infections are difficult and impossible to eradicate. Current models for biofilm research, such as micro-tilter plate-based models, the Cochlear biofilm device, the rotating disc reactors, and the microfluid device, produce biofilms on solid attached structures that are completely submerged in liquid media and do not include a combination of essential environmental parameters such as air-liquid interface, airflow, hydrodynamic flows. Therefore, they are neither optimized nor representative of in vivo respiratory environment. Consequently, with lack of understanding of biofilm growth in the lungs. Therefore, the objective of my study is to develop an adaptable device and set up a versatile platform to mimic the real biofilm development in the respiratory system and investigate the influence of aerodynamic and hydrodynamic on biofilm properties, and hopefully provide more some insight on developing novel anti-biofilm strategies. This diagram shows the platform set up for culturing biofilms on air-liquid interface we have developed. First of all, a dual-chamber device have been developed using microfluidic techniques, and this device is consisted by two flow cell layers made by PDMS and a porous membrane sandwiched between two PDM flow cells compose a dual-chamber structures that are able to establish diverse interface, and two plexiglass cover are for mechanical bondings. The membrane are interchangeable, which allows multiple purpose studies. An air pump is connected to the top channel to provide airflow, and the syringe pump is used to drive the liquid media flow in the bottom channel, and the waste were collected in the waste reservoir, and the bacteria were seated in the top channel and grew on the air-liquid interface. In our study, the biofilm have been cultured under both static and dynamic conditions for 48 hours. In static condition, the bottom channel is filled with static liquid media, with air in the top channel remained stagnant. And for dynamic culture, the liquid media was continuously supplied into the bottom channel, and the biofilms will grow in the top channel under the airflow and without the impact of airflow. The biofilm characters were studied in terms of its viability using viable cell number counts, and its morphology were visualized using SEM and confocal microscope. The permeability of biofilms were measured using pseudomonas permeability assay, and antibiotic suspectability to ciprofloxacin was tested by exposing the biofilm to a range of ciprofloxacin solutions for 6 hours. This SEM image shows that a biofilm cultured under static condition have developed a mushroom-shaped aggregates in its structure. And in contrast, the biofilms formed under dynamic conditions with continuous fresh medium supply have bacteria densely packed in the structure. And more interestingly, their surfaces are very smooth compared to static biofilm. The variable cell number counts result further demonstrate that the bacterial number encased in the biofilm structure formed under dynamic conditions was tenfold higher than static cultured biofilm. In these confocal images, the live bacterial cells are stained with green fluorescence, and dead cells are stained with red fluorescence. Comparing the biofilm developed under dynamic conditions, we can see that the biofilm developed without the impact airflow shows more dead bacteria on the surface than those cultured under the impact of airflow. The biofilm thickness measured from this image shows that biofilm cultured with continuous fresh medium supply is significantly thicker than static cultured biofilm. In addition, the airflow in the top channel produces thinner biofilms compared to when there's no airflow. The fluorescence permeability measurement shows a significant decrease in permeability compared to an empty membrane after had biofilms formed on it. In addition, the permeability of dynamic cultured biofilms was significantly lower than static cultured biofilms. Moreover, the existence of airflow further reduced the permeability in biofilm. The antibiotic suspectability test shows that after being treated with the same ciprofloxacin concentration, the percentage of surviving bacteria of static cultured biofilms was significantly lower than the dynamic cultured biofilms. And the minimum biofilm eradication concentration measured using our device for static culture biofilms is 800 microgram per ml, while for dynamic culture biofilms, this concentration is higher, which is 1600 microgram per ml. In conclusion, our work presents a novel dual-chamber microfluidic system capable of establishing air-liquid interface and controlling both aero and hydrodynamic flow environments as an in vitro platform to mimic the physiological features of a respiratory system. And our study have found that airflow has impact on biofilm structures, and due to structure difference in biofilm between static and dynamic conditions, there are differences in antibiotic suspectability outcomes, and dynamic cultured biofilms have lower permeability and more resistant to antibiotics. Overall, our study suggests that it is crucial to have a more physiological relevant to develop and test more targeted biofilm control approaches. This study was supported by Marie Bashir Institute and Macquarie University Research Excellence Scholarship. And I would like to thank all my colleagues in the group and for their support. And thanks for your listening. And I'm happy to answer your questions now.
Thank you very much, Xu. It was a very interesting presentation, and it's really exciting to hear the research which is going on to try and make the models that we're working in far more physiologically relevant. So we have had a few questions come through. We'll go through whatever we can within the time. However, those that we can't get to answer, we'll look to pick up offline. So one of the first questions that I've had come in is around, could you make a comment on the applicability of the findings in informing biorelevant models for respiratory pathogens in the form biofilms?
So my model are able to mimic the airflow in the lungs. Usually, the conventional models couldn't have airflow, couldn't have studied the impact airflow. And our model could introduce airflow into the during biofilm culture, and we found that the airflow has impact on biofilm structures. For example, it could, compares to those conventional culture biofilm, the airflow has produced the biofilm that has very smooth surface, and its permeability is lower than static culture and when there's no airflow. And also the antibiotic, because we for our current study we only test the liquid ciprofloxacin solution suspectability. So we didn't test the antibiotic suspect the difference, but we in our future study we're planning to test the suspectability on novel nanoparticle formulations. So we hope we can like detect the difference in nanoparticle drugs, because there are a permeability difference in the permeability difference between biofilms cultured with airflow and without airflow. Maybe the nanoparticles can level the permeability difference to increase to have so hopefully we can have a better biofilm eradication results.
Thank you very much, Zhang. So we're going to go now to Ben and Alex, who are in the suite below us. I think they've got a question or two for you, Zhang. Ben, Alex?
Yeah, hi. I've got a curiosity, and one or two questions have come in along similar lines, which is your system has a membrane between the liquid and the air, which you wouldn't find in the lungs. So how do you sort of compare the effect of that membrane to what you might find in the in vivo situation?
The membrane we choose right now is polyester membrane with the size of it's a porous membrane with the pore size of 0.2 micrometers, and it is a hydrophilic membrane. So when there's a liquid in the bottom chamber, the pores on the membrane will be wet. So we hope to using this membrane to mimic the lung system. So one side of membrane to mimic the blood flow, on the other side that with airflow to mimic airflow in the lungs. But we can also test with other membranes that like it maybe could mimic the permeability of the cell lines. In the future. Also, we can also use this model to have a co-culture model with cell line attached to the membrane, and also biofilms grow on top of the cells.
So do you think in the lungs that did the biofilm exist as like a raft floating on top of the mucus in contact with the air and the mucus, or does it grow under the mucus?
Grow on the in our group, one of our colleague have studied the biofilm grow on the mucus layer on the static air liquid interface, and it shows that the biofilm, the bacteria could form biofilms on top of the mucus layer within 6 hours.
And do you have plans to develop your model?
Alex, I'm going to just interject there. So thanks, guys, very much for the questions that have come through. We'll come back now to the main stage. Zhang, thank you very much.